Good morning. I'm Dr. Mindy Eichhorn, Assistant Professor of Education in the undergraduate and graduate programs.
And I'm Courtney Vitale. I'm a junior and a double major in elementary ed and math.
So before we begin our presentation today, we'd like to hear from you. All right? So, how would you complete this sentence with one word? Math is blank. So text your response, GCME to 37607, and then respond with your one word. Or you can go online, pollev.com/GCME. We'll give you about 30 seconds to text your response. Math is all right, so we all have an identity in regards to math, and our beliefs have been formed by past experiences. So whatever you texted has been formed by a past experience. Our research examined the math identity of early childhood and elementary ed majors and alumni at Gordon.
So why do we as educators research? To assist the teacher in improving and or refining his practices or actions, and to embark on a quest for knowledge about how to improve. As educators, it is important that we are constantly driving forward in improving our own practices. We are consistently striving to do better. And in order to understand what practices work or what we can do better, we must do research. Whether this be on a smaller scale in our own classrooms or on a larger scale, looking for larger data on curriculums and practices. Specifically, in our research, participants, which included pre-service teachers and alumni, tell their math story and how their math identity has been formed. The results from this case study are compared with information collected from other teacher prep programs of a similar size with surrounding school districts. So why was I interested in doing this research? Besides the initial thoughts that this was an amazing opportunity to learn about the process of doing research, as soon as Dr. Eichhorn and I started discussing the research, she brought up this topic as one of the many possibilities, and it immediately struck me. As an elementary ed and math major, I often get a lot of questions, basically summed up as, why would you ever do that? Which has caused me to question fairly often why people think about math often in this negative light, and how those thoughts affect their teaching and their students. In my own experience, I have seen a lot of intimidation surrounding mathematics, and I wanted to see how we can change these ideas in our students so that math does not continue to be something only smart people can do.
And why was I interested in doing this research? Well, I wanted to find out what we should keep doing, quit doing, and start doing in regards to how Gordon College trains teachers to teach math. Which parts of the coursework and classroom experiences have been most helpful? Do our pre-service teachers and alumni have a growth mindset in math? So we all have a core belief or a mindset about how we learn. According to researchers at Stanford, Carol Dweck and Joel Bowler, we could have a fixed mindset, believing that we can learn things but not change our intelligence, or a growth mindset, believing that with hard work and perseverance, you can learn and achieve at high levels. So you might have a growth mindset in everything except for math. I was also wondering, how can new teachers instill a growth mindset in their elementary students in regards to math if they have not experienced a growth mindset in math themselves? And also, because I am a professor, I'm still learning. I engage in reflective practice when I teach my courses, so I wanted to know how I could teach these classes more effectively.
So what did we do? We created a survey based on a couple of other surveys that have already been developed, which questioned participants about their feelings about math. We sent the survey out to the undergraduate, early ed, and elementary education majors, some recent alumni who are currently teaching, and the incoming freshmen and transfers. Additionally, we conducted interviews with survey participants, professors, and principals and math coaches at local schools. So over the summer, I spent a lot of time reading articles, books, and so on. I spent hours transcribing. I learned to use SPSS and to code interviews for themes. It wasn't quite what I expected, but I learned a lot. And what did we find? We found that strong math teachers, as described by students, are flexible, make connections to real life, encourage participation, support their students, and are passionate. While poor math teachers tend to give less help, rush through instruction, and solve problems in only one way.
And what should we keep doing, quit doing, and start doing in regards to how we teach math at Gordon for our teachers? So what should we keep doing in our assignments? We should have students engage in self-examination to understand and challenge their beliefs, attitudes, and actions towards math. And also consider how these attitudes will affect their teaching of math and their impressions of students. We should continue a focus on growth mindset. And we also found that participants noted their experiences in math content courses in college have made them feel more confident in math to teach it. We noted participants' perceptions of math before and after concepts of math, which is taught by the beloved Dr. Stout in our math department. So students and alum that have taken Dr. Stout's class believe that math is interesting and exciting compared to people who have not yet taken this class. People who have taken his classes acknowledge more of a growth mindset in regards to math. They remark that they feel smart when they're engaged with challenging problems, which fosters perseverance. Dr. Stout has created a safe environment for students to learn math and develop a positive view of this subject. In his class, students realize that with hard work, effort, and perseverance, they can learn and achieve at high levels. So, what should we start doing? We want to start addressing math anxiety and past math trauma through a math autobiography for our students and revisit it throughout the course. We want our students to ask themselves, what stereotypes do I hold about math? What is my math mindset? What do I believe about my potential as a strong mathematical learner? And what do I believe about my students' potential? What messages am I sending them about math? We want to highlight a safe learning environment and have our students reflect on why do they feel safe in these learning environments and how can they translate that into their future classrooms. So, how does all this relate to our faith?
As teachers, it is our constant goal to meet the needs of our students to enable them to learn. And as Christians, it is our goal to spread the gospel not only through word, but also through deed. This means that every student deserves everything we can give to meet their needs so that all students will feel that their ideas are worthwhile.
As teachers who are Christians, we want to show hospitality in our classrooms. We don't want to just welcome and invite students into our math classrooms. We want to honor their strengths, contributions, and accomplishments. It's important for us as educators to reflect on our beliefs and perceptions of math, since this will ultimately impact the way we teach math. We can be hospitable in our classrooms by offering equitable instruction and providing access to math for all. Each student deserves fair and just treatment in mathematics instruction that affirms their intellectual, cultural, racial, ethnic, and linguistic backgrounds. Equitable instruction does not mean equal, identical instruction for each child. It means that teachers must offer accommodations so that students have access to math. We aim for our pre-service teachers to be reflective practitioners, constantly evaluating their practice and beliefs. As Christian teachers, we can look at our unexamined prejudices that we might have towards math, even towards our students and their families. By focusing on biblical principles such as humility, grace, and human worth, our teachers have a greater incentive to meet the needs of their students in mathematics, in addition to meeting the requirements of academic state standards. So we would like to end with a question and a note of encouragement.
What about our research resonates with your experience in mathematical identity?
And how can you be involved in research as an undergrad? Since research is about finding answers to our questions, write down your questions and meet with one of your professors. Express your interest and ask your professor to mentor you in research methods this summer. Courtney and I actually discussed preliminary ideas the afternoon after last year's undergraduate research convocation. And just this week, we met with collaborators to work on a peer-reviewed journal article. So we'd like to thank the provost and the generous funders that allowed us to explore our research questions last summer. If you'd like to hear more about our project, we'd be happy to speak with you. Thank you.
Good morning. I'm Dr. Angie Cornwell from the Department of Biology.
And I'm Courtney Olbrich. I'm a junior biology major here at Gordon. We had the privilege this summer to collaborate with Dr. Lisa Spencer and Beth Israel Deaconess Medical Center in Boston, and we studied the role of eosinophils in inflammatory disease and how that relates, especially in inflammatory asthma.
So before you fall asleep from our title, I'm going to explain our title, hopefully in a fun way that will wake you back up again. First of all, what are eosinophils? They're a type of white blood cell, and you can think of them like one of the team of Rogue Defenders. How many of you have seen Rogue One in the theaters? A few of you? Okay, good. So you can think of them as one of these teams of defenders that are defending the body against attack. Now, what kind of attack are we talking about? We're talking about, since the 1940s, we've known about the role of eosinophils in defending the body against large parasites, the sorts of parasites that are causing disease like elephantiasis, horrible disease where these parasites are getting in, blocking circulatory system, sorry for the graphic image, but blocking circulatory flow and causing gross swelling of the limbs. So what are the eosinophils doing for you? How are they helping you? They are secreting chemicals. We understand they're secreting chemicals that are talking to other immune system cells and that are working to help the body by attacking these parasitic worms. For instance, attacking the outer coating, damaging the outer coating, helping to prevent them from multiplying, from laying eggs, and from growing, and so forth. And more recently, within the past 10 years, we've also learned about the role of eosinophils in defending the body against bacterial infection. So what you're seeing here, the eos are pictured in red and green, and you see them shooting out webs. This is like Spider-Man, another type of superhero, shooting out webs that are entrapping bacteria. So this is another great thing that they're doing for us. But I have to set the mood here, and it helps that I have a cold, so I'm gonna breathe. That's the best I could do. So what happens when these eosinophils go against us and go to the dark side? Some of you are afflicted with things like Crohn's disease, where you will suffer from severe abdominal pain after eating certain types of foods like raw nuts or like fruit with the skin still on it, or like greasy foods or caffeine or foods that have dairy or caffeine in them. And what you're seeing is an image of the inside lining of the digestive tract of such a person whose eosinophils have turned against the person and caused massive inflammation. So this is what we mean by the inflammatory disease part of our title. And so people with Crohn's, also some types of respiratory asthma, where essentially these eosinophils, we understand, they attack the body. And so if you're exposed to certain types of triggers, if you're asthmatic and you're exposed to certain types of triggers like seasonal pollen, like dust mites, or like pet dander, even some dietary triggers like raw peanuts, then what happens is, with exposure to these triggers, you get inflammation of the respiratory passageways, and you have eosinophils attacking the lining of these respiratory passageways, mucus buildup and so forth, constricting the passageways, making it really difficult to breathe. And so in some severe cases, you can even have death resulting. So what we understand about this pathway is that with exposure to some of these allergens, you get a pathway triggered where the eosinophils will leave the bone marrow and use the circulatory system to travel to the site of the invasion, and then leave the circulatory system, migrate into the tissues, and then you have all of those chemicals that they were previously using to attack large parasites and so forth. Now they turn on the person's own body and start attacking the person's own body. So what we understand is we understand what's going on, but we do not understand how it is going on. And that is the question that we will try to answer. So how can we strike back? Courtney is going to tell you about how we've been trying to strike back.
So there's a couple different ways that we tried to approach. There's about 3 different questions for our different assays that we're using. And the first one is, can we block these eosinophils from coming out of the bone marrow originally? Or can we block them from coming out of the blood vessels? Or if they do get into the tissues, can we block them from secreting those harmful chemicals that they're secreting that attack our own bodies? So, in collaboration with Dr. Lisa Spencer's lab, we're looking at a signaling pathway called Notch, which you can see is very complex. But it's important to know, because if Notch is indeed the intracellular pathway that's affecting this activation of eosinophils, we can look at ways to block it once we understand the pathway more fully. So you can see it's very complex, but you want to think of it as something, an initial trigger, like Dr. Cornwell mentioned, is going to trigger something in a chain of events, A, B, C, and D, which is eventually going to lead to something in chronic inflammation. In this case, we're looking at the inflammatory asthma. So if we're able to understand this pathway, then we can block the different points at A, B, C, or D, and then we're able to understand what is going on at these points. So how we're approaching this is to breed different groups of mice that are mutated or changed at these different points, so then we can better understand what's occurring in these mice and how it relates to us. So the idea behind a mutant mouse strain is that the mutant mouse is missing a key gene at one of these key points, A, B, or C. And then we can study this pathway more fully. So you want to think of each mouse that we're talking about as either missing one of those points, A, B, or C, in the events that lead to inflammation. So once these mice are bred, you can expose them to an inflammatory trigger. In this case, we're using house dust mites, to trigger the inflammation response, and then you can study both the progression and the severity of this disease in the different types of mice. So then you can look at what happens in a normal mouse that has all the normal genes and study what happens in that case versus a mouse that you've knocked out an important gene. And understanding both of these scenarios is going to help you understand which key genes are important in this inflammation response. And missing that gene is either going to lead to no inflammation response or hopefully at least a less severe response. So understanding these various responses are then going to allow you to understand the whole process of asthmatic inflammation even better. But as you can see, it takes a lot of work to breed these mice. And so it takes about 3 weeks for the mice to be born, and they're newborn pups at that point, and then they need to stay with the mother for another 3 weeks until they're weanlings. And at that point, they can be used for experimental use or breeding. And it does take a lot of work to breed these mice, and so it's important to understand that while we don't have time to go into the genetics of breeding, the offspring are not going to have all of the mutations that we're looking for in the parents, only a portion of them. So this left with a question of how to see which offspring did inherit that mutation, and this is done through DNA testing and genotyping. So here you can see the process that I went through many times this summer to test the genotype of the mouse and see if they carried that mutation that we're looking for. And so you take a small piece of the mouse's tail or a tissue biopsy, and then you're going to extract that DNA from that tissue and amplify it and make several copies of this gene, so that when you run it on a gel, as you can see mine here, you get a concentrated band of this amplified DNA. And so you can see in lanes 1 and 2, there's just one band, so that means that it's a mutant mouse and it has 2 copies of that mutant gene. Where in lanes 3 and 4, there's 2 bands, so one of the copies is the normal type of gene and one's the mutation, and then finally lane 5 is just 2 copies of the normal gene. So this DNA genotyping had to be done for all of the mice until we can find parents that are only going to produce one type of offspring. But because no one has ever bred some of these type of mice, we're not sure exactly what was going to happen to them when we change their genes and what happens to their growth and development. So here you can see I'm holding two mice that are litter mates and should be the same size, but one is considerably smaller than the other. So this is something that we're still tracking and looking into the statistics about what happens when we take out these genes and how it affects growth and development, because that's just as important to understand. But after we get the right mouse and we characterize it and make sure that it's healthy, we culture that bone marrow that we talked about originally. And here you can see another picture of those eosinophils once they've been in the bone marrow and they're cultured in a cell culture. So we also this summer worked to create a double mutant mouse. And this is something called an EO-Cree mouse that's crossed with an MTMG mouse. And the MTMG mouse has tissues that are selectively fluorescent red, and you can also turn on a molecular switch inside these cells and turn them to green. So this is really important because then by color, we can make sure that we're looking at and testing and experimenting on the right type of cells. So here's a picture of what that looks like fluorescently on the tissues in both red and green. So on the left, you can see the tissues are red, and then those same tissues, when that molecular switch is turned on, are going to be green. Here was a study showing that this can be also done selectively, and a group of scientists did this with liver cells, and you can see just an example of both of those colors being represented in the same tissue. So here's some of our unpublished data, and this is what happens when you cross that MTMG mouse with the EO-Cree mouse, and both of those double mutant mouse together give you a mouse that only the eosinophils are going to turn green and not the whole tissue. So here on the left, you can see that the MTMG mouse tissues are red, and then the green arrows are pointing to eosinophils that are green on the right. And so this is incredibly important because at any time in our study, we can look at cell culture or look at tissue sectioning, such as here, and you can see where those eosinophils are, and that will help you understand both the severity and progression of the disease like we're looking at. And then a final point of the importance of the fluorescence is that this can be used in cell counting. So it's really important to know the ratio of green to red cells that we have. And so with this fluorescent laser, it can decide whether the cell is green or red, and then it can count so we can see the proportions in the different types of mice that we're getting that eosinophil response when it's exposed to a trigger. So this is just a small portion of our research and what we've been working on and what we continue to hope to work on. And I think I just want to hope that it comes across my passion for medicine. And I've always been interested in medicine, and through this process, I've really experienced my passion for clinical research as well, and that ability to alleviate suffering through this type of work. And so I just hope that it encourages you to get involved in whatever you're passionate about, even if it seems far stretched. Like I didn't expect my freshman year that I probably wouldn't have believed if someone had told me that I would be working on projects with Harvard Medical School, but it is possible, and you have to follow that passion, whether it's research or something else. So I encourage you to just follow what you've been called to do and to do it well.
So in closing, I just want to leave you with a final thought. Some of you may have been looking at these data thinking, that's all well and good, but I could never do that. And so I want to leave you with the words of a wise master who said, "Oh, master, more times failed than tried, ever beginner." And so by no means do I want to imply that we are masters, but we have been working all summer, troubleshooting, optimizing, and the results that you saw from Courtney's work was the result of many, many times of trying something and doing it over and over again. So we want to encourage you, no matter what your field is, no matter what your major, there's research out there for you to do. And we want to encourage you, start this journey, travel it, fall down, get back up again, try again, and you will learn more along the way than you ever thought was possible.
We would like to close in thanking just the provost for her ability to give us this time to study this and follow our passion in this clinical research, and then also to Dr. Lisa Spencer, who's our collaborator at Beth Israel, and then a list of all the other people that have helped us through this journey. Thank you.
This is not product endorsement, but we realize that the cosmetic product EOS, which is our cell, eosinophils, is also shaped like a Death Star. So we would like to personally thank the provost for her sponsorship of this and share with her